No, press it. Yeah. 11 o'clock on the first Sunday of the morning means it's time for Chai and Why at Prithvi Theatre. Thank you all for coming. Thank you all those who have joined us online and are watching this. Before I introduce the speaker, you can see that there's a microscope out here. So if you've come, there's something extra for you, right? Sorry, online audience, you will not get to peer through the microscope, but hopefully you will still enjoy the rest of it. Now, if you're watching us online, please remember that the only way we can interact with you is through the chat. So please put your questions and comments in the chat. And we have Sandhya students are here who will monitor the chat, and we will take those questions up. Of course, here you can always raise your hand and ask us something. It's perfectly fine. Request to the speaker. During the first part, if somebody asks you a question, please repeat it for the online audience.
Yes.
Okay. So this is something which promises to be a very interesting talk about something to do with a worm and Nobel Prizes. Okay, not just one, but 4 of them. But Sandhya is going to tell you the story. But let me just formally introduce the speaker. Sandhya is a sort of regular at Chai and Why. She's done many sessions here before, but still a faculty member at TIFR, a cellular neurobiologist is what she calls herself. Did her PhD at Brandeis, then went to the School of Medicine at the Washington University in St. Louis. From there, came back to NCBS, the National Center for Biological Sciences. Then joined TIFR, where she works on worms, what she's going to talk about. And one thing I will say is, she is one person who very seriously takes the fact that in TIFR, which is the Tata Institute of Fundamental Research, there is some fun to be had. Okay. So she is the one who makes research fun, fundamental research. And I think you will enjoy the journey that she will be talking about on this tiny little worm. So without further ado, hand it over to you. And please remember to ask questions. We will sort of do this first part of the talk. Then we go out for chai. We will not live stream. You come back and that we can ask a lot more questions. That's not going to be live stream, so don't worry. You can ask all your questions over there as well. But right now, let's go ahead. And online audience, please remember to put your questions on the chat. People over here, please put your phones on silent. Thank you so much.
Thank you, Arnab. And thank you all for coming on a Sunday morning. I usually have to drag myself out of bed. It's all on. I have to usually drag myself out on a Sunday morning. And it's really nice to see that some people perhaps don't struggle as much as me. So one of the things I'm going to talk to all of you today, including the online audience, is something I have been in love with for a very long time, which is the little organism that I work on, which is called C. elegans, or the worm, as it's called in a friendly manner. This little drawing that you're seeing over here is actually a student in my lab who does art as well as science, did that little illustration to show that the worm can be used as an investigative mechanism to understand fundamental problems in biology. When I talk about this one tiny worm and what it does, I will touch upon the culture of doing research among the people who use this organism. So what I told Arnab earlier and what I will tell you today, it's a little science light, which is I think probably the first Chai and Why I'm giving, which is a little science light, but also has stories in it. And I hope you enjoy it. I think in this day and age, across the world, whether you are in countries which are well funded for research, or countries like us, which don't even reach 1% of GDP for research, compared to, say, Israel or South Korea or even China, the space that you have for doing curiosity-driven research or just figuring things out for fun is often shrinks. I hope through this lecture, you begin to see the value as the general public who supports most of the research in this country, the value of just letting your curiosity drive where you go in your science and have unexpected rewards. Okay, so that's the message that I would like you to take home today. The worm and the people who work using worm share this culture of curiosity, but also a culture of openness where we share a lot of information with each other and there isn't unhealthy competition most of the time. So I'll show you a little picture, which is you take this little nematode, which is 1 millimeter long. So it's really tiny, 50 microns across. We'll show them to you afterwards. And if you hold it up to the light, you'll see them as little wiggly lines that are moving around. Okay, so they're kind of tiny, sort of visible if your eyesight is reasonable, but you can, of course, see them really well under the microscope. But if you take a scanning electron microscopy, that is, you take this little worm, lay it on a whatever surface, and put some stuff on it, and you set it up in a high resolution microscope, this is exactly what you see. It's a beautiful worm with its little tiny tail. It's got a very simple morphology. So people have been using nematodes for a very long time in research, from the 1800s. Biologists is a curious bunch of people, will sort of ask questions where it was possible to understand. And then they will take that particular model organism forward. So there was some work done with horseworms to try to see if chromosomes were important for, and I think all of you have heard of chromosomes? Yeah. So chromosomes were important for heredity. So there were people looking at those kinds of questions. And soon they realized that some other kinds of worms were better, that they isolated more worms from the soil. And Caenorhabditis species of worms, several were isolated by various investigators in the UK, in France, and even in the US. And people started having struggles because you can isolate something from the soil, but you have to figure out how to grow it. And often that was just an industry in itself to figure out how it grew, what it did, what ecological niche it lived in. While there were people using C. elegans in this manner, there was one very fabulous and wonderful scientist called Sidney Brenner, who was in Cambridge, working in LMB, which was headed at that time by a very famous physicist called Max Perutz. And they were figuring out with many other peoples, including Watson and Crick, you might have heard of them, for the double-stranded structure of DNA, etc. A lot of things about how life works. That is, you have DNA. From this DNA comes RNA. From the RNA, you have translation taking place, and you have a triplet codon, which is read in the RNA to give you one protein, one amino acid, which attaches to another amino acids to finally make a protein. So in this exciting time, where a lot of scientists were present in LMB in very scrunched, tiny little spaces, not large bays that you might see in many labs now, people were figuring out fundamental principles of how cells worked. Laboratory of Molecular Biology. Okay, so there were major discoveries in biology, and Sidney Brenner, who was a South African who had come to Cambridge to work and to do his PhD and then to do research, was part of that enterprise, a thriving, intellectually fertile place. Lot of those problems were getting solved. Sidney Brenner was very, very smart. In fact, I have met and had tea with him. I've met him more than once, and I had tea with him, and very amusing person as well. And he felt we have sort of understood how cells work. You know that DNA gives you RNA, gives you protein. And he asked the question, what are the big problems we need to solve? And then he went and spoke to the director, Max Perutz, and tried to convince him to let him do whatever he wanted and to give him money. Right? It's very important. His thought was that the big questions which needed to be addressed in biology were development and understanding how the brain worked. Development is a process where you have a single cell, which is the egg, which gets fertilized and then gives rise to this entire organism. Every species goes through that. You start with a single cell and you stereotypically get the same organism. So every human embryo, it's a fertilized embryo, will grow up to be a human. It will not grow up to be a tadpole. So it was clear that there must be steps, what he calls unitary steps of this ill-defined process which needed to be studied. So he came up with this idea and he said the other big frontier is the brain. We need to understand the brain. I'm going to take a step back and say, can any of you tell me why you think the brain should be studied? Why bother? Just tell me what you think.
Because it controls the whole body.
That's one possibility. Anything else? Who has aging grandparents, parents? Man, I don't remember phone numbers anymore. My parents certainly don't. Right? So, I mean, just you see that there is change, there's aging, there are changes in how the nervous system functions through age, and there are, of course, diseases. So there are very good reasons, fundamental reasons as to how does it control speech, movement, thought, all of that, right? So these are big questions. So he said, if you want to understand these unitary steps of the process, we need some sort of model system. So at that time, a model system is basically an organism that you take and you say that this is going to model everything that we do in every, if I figure it out in this little simpler model system, I figured it out for a human being. Bit of a stretch sometimes, but sometimes not. Okay. At that time, people were working on fruit flies, which was Drosophila. But if at all you look at the diversity of species and organisms which are around us, you obviously want to look at more than one organism. And he felt that the fruit fly was quite complex. And he said that he wanted to pick something extremely simple to start working on these questions of development and neurobiology. So let's look at some worms. Not under the microscope, but right here, you can see them wiggling around. These are middle-aged worms moving around like little tiny, some people have told me like snakes. They're very cute. They're transparent. I'm very fond of them. So when I asked you the question, what is an approach that most people take to solve questions? We first ask, why do you want to study this question? Not why does this process occur the way it does, but why do you want to study this question? You guys gave a beautiful answer as to why we need to study the brain, just so that we understand ourselves and understand movement and other things that organisms do, as well as for disease. Next thing is you want to know, how are you going to study it? And how are you going to figure anything out? And finally, what have you learned? And that's the little journey that I will take you through. And while I take you through that journey, I want to tell you that scientists come in many flavors. Okay, scientists come in many flavors. They are people very different from each other. Some are people who will huddle on a microscope and not want to talk to any other human being. Other people are gregarious, work in big groups and are very happy doing that. Some are collectors. They just want to go out into the nature and collect a lot of information or in their own lab. Some are classifiers, tidier-uppers. And this is a Nobel laureate who's describing the various kinds of ways people approach problems, scientists approach problems which they feel comfortable with. So what Sidney Brenner did when he started working with worms is he wrote to someone called Doherty, who was at that time in California, and asked him for some worms, and then set up a culture which persists to this day. So Ed then wrote a letter to Sidney and sent some worms and sent him all the information that he had to grow those species and said what the problems were when he was trying to grow that particular species. So that's a culture of openness and open sharing, which Sidney then again propagated in his lab and from the many people who emerged from his lab. So what he knew at the time when it started, approximately, but he sort of nailed it down over the next few years after getting the strain from Ed Doherty, is that you have a lifespan which is very quick. It takes 3 days to go from one adult laying a little egg, going through various stages and becoming an adult again. What's the big advantage of that? You can do experiments really quickly. If you're going to do an experiment with a human being and you want to have kids come from human beings, you have to wait till they are at least 18 now by law. But certainly you have to wait well over a decade before you can have any progeny at all. Same thing with mice, you have to wait a really long time. But worms aren't like that. They are fast, and therefore you can get a lot of information and do your experiments very quickly. So he said he had this organism, and his idea was very simple. And this comes back to how do you approach the problem? Okay, let us say I give you, all of you have seen a cell phone and a remote or whatever is there. I give it to you and tell you, I want you to set it up that it doesn't work. And I want the other person to figure out why it doesn't work. What is the first manipulation you will do? You will remove the cell, okay? That's one thing. What else will you do? Pour water in it. It will never work after that. Pour water in it, it will never work after that. What next? Huh? Put some virus in it. You guys really don't want it to ever work again. How about just turn it off? Right? So what are we doing when we try to understand how a system works? One set of observations is just observe it and see what is happening. The second is to do a manipulation, pouring water, turning off this, removing the cell. All of that is a manipulation, right? So the way you do manipulation with an organism is to feed it a mutagen. It's the equivalent of exposing yourself to a nuclear bomb blast. So what do you do when you feed this mutagen or something which will change the genetic code and therefore the RNA that comes from it and therefore the protein that comes from it? You can potentially make a change in the organism. So you do the equivalent of a nuclear bomb blast, like happened in Japan, where you saw that there were very many birth defects, right? And then you go back in your little C. elegans culture or worm culture and see what are the changes that you see in the progeny. And you select them. So implicit in this is the assumption that if you do such an experiment and change the DNA, you will be able to get mutants, which you will be able to pick. It's possible to get a mutant from the outside. Nothing is visible to you. After all, worm can't come and talk to you, "Mera sar dard ho raha hai." Ye to nahi ho sakta, right? That's the big advantage of having humans as a subject for research. They can tell you what they are feeling and thinking. Worm can't. So if the worm doesn't physically look different or move different, you can't spot it, right? So the idea was Brenner did not even know when he started this work if he would be able to get mutants. He had to figure out how many chromosomes there were, whether you could get mutants, whether he can get multiple mutants in the same gene which encodes a functional protein, all of that, right? But he worked out all of those things. Here are some examples of mutants he got. This is a normal looking, and this is from his paper. This is a normal C. elegans. I showed you little wiggly things which were happening. This is dumpy, that is short as well as fat. Okay, this he called small, which is only short. It's not fat. Okay, they should have had a normal size fat also to describe me. And then this is long and thin, right? He also got mutants which don't move properly. And what would you say of things which don't move properly? One possibility is that your nervous system doesn't work well. Another possibility is your muscles don't work well, right? But now you see you're beginning to touch closer to the question of neurobiology. And by looking at these mutants, you're also getting close to what decides animal size? What decides animal shape? Does it happen from the way it develops? So the whole range of mutants that he got, which he classified, several of them are still being worked on. So nothing he did has ever been wiped out. We still look at, so the alleles that he, or the mutants that he called, are called E with some number, okay? We still work with many E and some number mutants to this day. So tremendous contribution to the entire field. Okay, so we now knew at this time from Sydney's work that you could get mutants, you knew the chromosome member, you could get behavioral mutants, you knew a fair bit of stuff. What is the other piece of information you need if you want to understand such questions? This was an experiment done thoughtfully and was extremely meticulous. So John Sulston, who was also in the LMB, decided that what he was going to figure out is lineage. And I'll explain what lineage is. You start with one cell. It becomes 2, it becomes 4, because each cell divides into 2, right? So it increases exponentially in numbers. And then you go on to see how many cells you get in at the end. And you figure out each cell gives rise to which kind of cell. So in the sense, in the adult animal, you can map every cell to the original first embryo single cell, which is really a tour de force for development, has not been done in any other system so far. It was done here. It took a long time to do. Essentially, you look at one embryo, or one animal, do image it and keep watching it and drawing on it for several hours. Then you put it in the fridge overnight to pause it. Next day, you take out the same slide, put it on the microscope, and continue the experiment. Right? And you have to do multiple things. So he started, and this is John Sulston. He did all of this stuff in the embryo, and he also did it in post-embryonic, to how the embryo becomes an adult. So for instance, you start with a single cell, you become a folded up worm, you hatch, and then become a worm which grows up and increases its length, and so on and so forth. And he made these kinds of trees, saying you started with this cell, it divided, and he gave numbers to every cell that came, and this is one of his drawings. When he was doing this meticulous work, which he thought he would understand something about, development of the organism, he found that 131 of the 1,090 cells that were born died. Hitherto, nobody knew that such a thing even happened. This was a completely new phenomenon. Were they trying to find a cure for something? No. Were they even trying to find some answers to something? No. This was pure observation and serendipity and careful experimentation, where he spotted that cells which are born and look healthy undergo death, and the same cell will undergo death in every individual animal you look at. So it's programmed cell death. Bob Horowitz worked with John Sulston. They were all part of this LMB group working under, technically, Sidney Brenner, because although I've heard from people who worked with Sidney that he spent most of his time motorcycling around the countryside in England, and he would show up once in a while and discuss science and go off again motorcycling. In fact, one of the people in the lab I've heard spent the most time talking to him when Sidney had fallen off the motorcycle and broken his thumb bone and was laid up, so different times. So Bob Horowitz and Sulston worked together to identify, taking that same mutagenesis approach that Brenner had established, to find out if I take out some particular feeder mutagen, now does cell death stop? Can I find the death genes, right? So here's an example from Bob Horowitz's Nobel Prize lecture, and you see that these are the cells which are present, which haven't died, and in a normal healthy animal which is not mutated, those cells will die. So using these approaches, they built this entire pathway of what was the genetic program, is which gene acted first, which acted second. None of the details matter, but you know, anybody who works in biology will draw arrows. Sometimes it's not clear what those arrows mean, but A happens, then B acts, then C acts, then D acts. So you had all of these where you have the cells born, then you decide which cells are going to die, then you actually kill them, and then you do something else. And then you have to finally clear them so you don't have any mess and fuss around. That's what programmed cell death is. So this seminal work showing that you could make mutants and get a variety of mutants, that you have a cell lineage which is fixed in which cells die, and you discovered a new process called programmed cell death, and that you had genes which control us, death genes which were present, all led to the first Nobel Prize for the worm, given in 2002 to Sydney, Bob Horowitz, and John Sulston for their discoveries concerning the genetic regulation of organ development and programmed cell deaths. And if you think this is some worm-specific thing, absolutely not. In all of us, and in rats and mice, many more neurons are born than finally form part of a circuit. Those extra neurons die. So young babies will have often a larger number of neurons than slightly more grown-up kids, simply because those cells did not meet their targets, and so they basically undergo cell death. In cancer as well, you see cell death taking place. So there are many biological contexts in which if you want to remove some cells, you turn on this pathway and turn on programmed cell death. So through this serendipitous observation, they uncovered something which is fundamental to biological processes. Brenner in his award lecture says, without a doubt, the fourth winner of the Nobel Prize this year is Sinorhydritis elegans. It deserves all of the honor, but of course, it will not be able to share the monetary reward. While we are thinking of Nobel Prize related work, there's lots of work which has gone on in C. elegans, which has not ended up in Nobel Prizes, but has made seminal contributions to fields, important fields. For instance, there were several scientists, one amongst them is Cynthia Kenyon, who actually discovered the first mutants which led to long-lived worms. You know, worms which lived, I think, 3 times the length of what were. Normally, they will die in a month or so. These lived much longer, and they were just not decrepit worms that lived much longer. They had improved health span. And therefore, one area of research which worms have provided a lot of biological insight is how aging occurs and how that can be, sort of have healthy aging. The insulin signaling pathway, dietary restriction, all of them have been observed in worms as well. So again, I will emphasize, while every piece of work, major piece of work which is done using this model system may not end up in a prize, fundamental insights have come from working on this organism. But I'll talk about one thing which I think many people have heard about. Have you heard about genome sequencing? We know all the human genome sequence. People will say, oh, we're going to get the tiger genome, we're going to get the pig's genome, something else, right? People sequence that all the time. We are going to get 10,000 Indian humans' genome. The first multicellular eukaryote which was sequenced was C. elegans. And it was sequenced, there was no hypothesis driven. They said, no, we're going to do it because if we do it, we'll be helping the community. And they came up with more and more reasons. No, but then we will know all the genes in an organism. The initial thing was, if we had this, we can find out which genes are mutated and we'll be able to figure out biological pathways. But then, once they sort of got into it, they realized there'll be a lot more rewards coming from it. The two things that came, right? First, it was the first multicellular eukaryote that was sequenced. It took a consortium approach where it was sequenced in different continents by different teams which openly shared data. This was the time when Craig Venter and others were saying, we are going to patent some of the human genes, we are going to sequence the human genome and keep this information only for the people who do the sequencing. And the worm community, John Sulston, Bob Watterson, etc., loudly and vociferously pushed to make sure that the culture was one of sharing, and it was sharing with the public openly, which allows all of us, anybody who wants, to look at that information. Okay, that was a huge win. And while they were doing that, because it was the first larger genome being sequenced, which was not E. coli like a bacteria, you had to develop various technologies and various platforms, software platforms, which were new, and that really contributed to ensuring that the human genome could be sequenced. Okay, so there were several good things which came out of the fact that the worm people got into the game early and carried with them the culture of openness and sharing, which they now translated to efforts which I think are much more important for public benefit. While this stuff was going on, and you see that it was sequenced in the end of, it was published at the end of 1998, it was very exciting for people like me who were just starting out in worms at that time. There was another piece of finding which came, and the 90s were an incredibly fertile period. So there was a group. So let's go to the next slide and then I say, you know that you have DNA in the nucleus. From the DNA, you have RNA coming, and then the RNA comes out of the nucleus and then will make protein. People had been playing around with this idea that perhaps you can prevent this RNA from becoming a protein if you put a sequence in which could hybridize with that RNA, because it's a sequence and you can have complementarity. It was called antisense RNA. And so, of course, more people were interested in saying that let's do this kind of experiment, let's feed the animal or inject the animal with antisense RNA. So you have a particular piece which you think is the RNA which will give rise to protein. You put something complementary to it. It's the antisense RNA. And then you'll want to do a control experiment. And your control experiment will be the same sequence as the RNA of the messenger RNA, which will give you your protein. So Ken Kempfuis, and this is in his paper, showed that whether you put the sense or antisense RNA, you were getting, the embryos were dying when it was against a particular gene. They didn't make any comment on it. They just wrote in the paper, we don't understand it, and left it at that. Andy Fire and Craig Mello, one in Carnegie Institution, where Andy Fire was also developing lots of reagents for the community. So he was not just doing research on biological problems, but he was developing, vectors and so on for the community. He said, oh, let me play with these experiments. And these are the experiments that these people did themselves, okay? They didn't have an army of people in the lab who were doing it. So they started injecting sense and antisense independently, they were doing it very carefully, making sure there was no mixing up between the sense and antisense, and they were injecting it. When they put both pieces together, putting double-stranded RNA. And here's a piece of data from them. You just did sense, nothing happened, and this was very carefully done sense. You just did antisense, nothing happened. You put sense and antisense together. He's putting a double-stranded RNA together. It suddenly silenced gene expression. And this very nice paper that they did really well, it's a really wonderful paper to read and a lot of detailed data. In fact, there's only one example. That table is exceptionally long with, I think, 7 or 8 genes which they have tried. They went on to call this RNA interference. Why was this important? Let's come back to Brenner. What did I say about the cell phone experiment where you pour water into it or remove you want to break something figure out what you have broken to figure out how the process works. Now, if you're doing an equivalent of a nuclear bomb blast, you don't really have control over what you're changing because so many things in the DNA will change. What this allowed you to do was to say, you have the whole genome sequence now. You know every gene and every protein which will come out from your genome. Now you can do RNA interference against specific genes which you get from the genome and figure out a function. Enormously powerful, useful in every organism. Therefore, you can study something in plants, you can study something in Drosophila, you can study something in cells that you've pulled off from your epithelia and grow in a plate. You can figure out in any place. And nowadays, people are using this kind of technology to help with knocking down genes which are mutated in patients. What they realized, and they didn't know the mechanism at that time when they published it, what they realized is providing this double-stranded RNA against specific genes silenced them. And that led to the second Nobel, which was given for work done in this little model organism. So Andy Fire and Craig Mello got it for gene silencing by double-stranded RNA. And this universal means to interrogate gene function by reducing gene expression, right? So immediately it transformed biology. Everybody started doing things like, we're going to knock down our gene of interest, what we think is a good candidate, but you also got groups of people saying, we're going to knock down every gene in the organism and look at some particular process, maybe development, maybe how the animal moves, anything. So you really got a mine of information from all of this. Okay, so obviously you're using this model system, and at that time, you're making progress in how to silence genes, you'd figure out a new biological pathway, but biology and many kinds of science is actually held back by the kind of technology that you have. I'm going to talk about another piece of technology where the worm played a star role. Okay, and that's called green fluorescent protein. Here's again, and as I said, both Andy Fire and Craig Mello or Ken Kempf did not go out trying to figure out what double-stranded RNAs were doing. They did a careful experiment, noticed something, and say, oh my God, we're seeing something very puzzling here, we should follow it up. Right? It's a very different way from saying, I want to study how we stand upright. It's like, oh, I saw something interesting, let me go check it out. It's a very different approach to doing science. So again, with green fluorescent proteins, if you have ever gone to any, I have not seen jellyfish in Mumbai. I have seen them in Goa. You see these fluorescent little things in the night. People were curious. So Shimamura was curious as to why is it fluorescing? Let me find out what is causing that fluorescence. So he found some protein, or what he thought was the protein. And somebody else, Doug Prasher, kind of cloned it, found a little piece of DNA which might encode it, etc., etc. There's some very nice stories over there, but I will stay away from them. So Marty Chalfie, who was in Colombia, was hearing a seminar neither by Shimamura, who was in Japan, or Doug Prasher, who was almost on his way out from science at that time, but somebody else who was talking about possibilities of labeling things with fluorescence, so on and so forth. And Martin got very interested. He said, I want to do this. So he got a rotation student who was in the lab for a short time to basically express this little piece of DNA which made GFP in C. elegans, using whatever promoters that he was using, whatever things to drive it. And this is the first set of pictures he got, where you see this lovely, beautiful green neuron. You can see that this green neuron is growing, and you can see how it is protruding. That's why it's not yet made its communication hubs, the synapses, and something else which is all grown out. And this really transformed research. It's not as if people hadn't tried before. There were some technical reasons why he got it to work and others didn't, okay? But he got it to work, and this has transformed biology, completely transformed all areas of biology. Other people, notably Roger Chen, made and identified a lot of these fluorescent proteins, which come in a rainbow of colors, and I'll show you a movie. And of course, in worms, this is a transparent living animal. You've not killed it. What it allowed you to do was to look at cells and look at proteins in living organisms in real time. No longer do you have to sit there and wait days to figure it out. So, oh, this protein turns out now, and what does it do? Things became much easier. I can't emphasize enough how much it has transformed biology. Our entire department, majority of the people use approaches like this combined with microscopy to track them. So let me show you a couple of images from our own lab. Here is a little worm, which is in a little microfluidic device. We trap it, turn on UV light. You have a protein which is tagged to the GFP, and you see protein move in. They are present in some membranous compartments. This cargo moves in both directions. Sometimes it doesn't move. Excellent. This is a few minutes movie, and we can see exactly what is happening. This is inside the axon, which connects the cell body of the neuron to the place where we'll talk to another cell, typically another neuron or a muscle. You can now take two different colors of these fluorescent proteins. And again, I'm going to show you a neuron. And then you see when they're present together, it's yellow. When they are separate, they are green and red. And I'm looking at these two proteins and how they are present together. And they actually mark organelles and how we expect them to separate. And here is, you can see very nicely the green, red, and the yellow moving around, right? It's incredibly powerful to be able to track fate of organelles, fate of proteins, and fates of cells. And now the entire area of cell biology opened up for everybody in this world. So what you got was a Nobel Prize of Chemistry for the discovery, for the development of the green fluorescent proteins. But Chen went on to develop a whole rainbow of colors for us to use. And that was, of course, transformative. I come now to the last Nobel Prize, which came to C. elegans. And I'll first describe it before telling you how they got there. As I said before, you have DNA, which gives rise to messenger RNA or RNA, which comes out in the cytoplasm and makes protein. As I said with GFP also, Shimamura was just looking for something which made GFP. Other people picked it up and said, oh, this might be useful. Marty just did a crazy experiment and it worked. So a lot of serendipity, no great plans, and just curiosity carried the day. So I'll come to this again. So, yeah.
About last Nobel Prize, cancer.
With the help of fluorescent proteins, can you detect cancer? That's not typically what is done. But if you're looking at, supposing you know that there's a particular kind of cancer where you know certain genes get upregulated, then you could try to develop an assay based on that. But that's typically not that's done. It's not done because the human being who has cancer will have to carry the GFP containing protein, which we don't, of course, normally express, right? It's only present in jellyfish. So there are other techniques that we use to detect cancer.
Cells which are biopsied and then culture them and then put the GFP in those and then see how they're divided.
Of course. So the question is, can you biopsy some cells from wherever, whether they are cancerous or not, and then introduce GFP? And the answer is yes, and people do those experiments. And they will do those kinds of experiments to see if something, something about the fate of the cell, typically. Is it dividing faster? Is it dividing slower? Maybe if you can make a fusion protein, whether it's localized the same way as in a normal cell, yes, you can do that. But that's after the fact, right? After you've taken it out.
The cancer.
So typically cancer is change in rate of cell division. So it's uncontrolled cell division. So you may not need to use this kind of technology to identify whether your cells are dividing very fast. But not that it cannot be done, but it may not need to be done. Because all you have to see is whether you have a large number of cells. Is there any other question so far? I'm going really slowly, but we'll be wrapped up soon. Green fluorescent protein. I'm sorry. What is GFP? Green fluorescent protein. And then you have red fluorescent protein. And then you have tangerine, scarlet, cherry, whole version. All kinds of versions. Things which you can turn on at this particular wavelength. There's a huge number of variations, right? So very useful. Anything else? Anything online?
Can gene regulation and microRNA help in the cure of cancer?
I will come to it because we have not talked about microRNA yet. RNAi and microRNA are somewhat, they use sort of, there are similarities in the pathway. What we've looked at with RNAi is you can turn down gene expression. So if your cancer is caused by just one gene getting upregulated, you could try to do RNAi for it. I can tell you one example which I am familiar with, which is for a neurodegenerative disease, where there is a mutation in the motor, which moves cargo around. So it's like a little car which moves cargo around. And what they do is give a single-stranded RNA, which is antisense, which complements that mutation and drives just that RNA to get degraded. So these patients are they have mutations in this gene called KIF1A, and this is a rare disease, so KIF1A-pathies, and there has been significant success with this, where if they have mutated gene, they have sometimes 100 seizures a day. And by treating them, they're very small ends, of course, treating them, very small number of patients who have been treated, but very small number of patients are treated, but have been treated such that over 100 seizures a day come down to about 20 to 30, which is a big win for the families and for the patient. So that is one approach which has been taken with RNAi. Okay, so when we come to, please tell that individual, I will come back to microRNA. Okay, so what Victor Ambrose and Gary Ruvkun, who were both postdoctoral fellows in Bob Horowitz's lab, who was one of the Nobel Prize winners, were studying questions of development, like how do worms grow, how do they develop, so on and so forth. And I'll just tell you the bottom line. They found that one tiny RNA, okay, which the cell itself was making, this tiny RNA, which is 22 nucleotides, bound to a proper long messenger RNA and was able to change gene expression. So I'll just tell you the steps which led to this discovery of what they then called microRNAs, or RNAs that did not give you a protein, called non-coding RNAs. Okay, this was through the awesome power of worm genetics and genetic screens. I'll not go into too much detail, but because this is something that I really love, I want to share it, even if you don't understand it fully. I'm very sorry. It's like, I can't let it go. So LIN4, lineage 4, did not make a vulva. Vulva is it's like the vagina. That's where the eggs come out. So if the vulva is not formed, the eggs grow inside the animal, the animals will hatch inside the parent, and then they will all come out. Okay, they'll eat their way out. So the mother sacrifices herself for the baby. Then somebody who was a PhD student in Bob Horvitz's lab, I forget his name, I think it was Reitman, who looked for a mutation, and there was with another gene, Lin14, which did something different, which also did not give vulvas, but it could revert, you could mutate it, and with what in technical terms you'll call a suppressor screen, and you could get some change in that gene, which now allowed it to function. They also saw that the timing of cell division in both Lin4 and Lin15 mutations was changed and was opposite. One was faster, one was slower. And when you put them both together, they came back more or less to normal. Then they got another particular mutation in which Lin14, which usually made a vulva, now did not make a vulva and looked like Lin4. So finally, after getting all of these alleles, and I should say that the first Lin4 mutant that they got was isolated by Padmanabhan Babu, who was a faculty in TIFR Mumbai when he was working with Sidney Brenner during a sabbatical. And he is acknowledged as such for getting, which was a very central thing to help them figure out what Lin4 was. So the first thing they figured out, Victor Ambrose figured out in his lab that Lin4, and took them a really long time because it was completely new, that Lin4 gave this small piece of RNA, which was a non-coding RNA, which did not give protein. And meanwhile, Gary Rufkin, who was also in Boston area, because they had both left Bob Horitz's lab and started their own labs, where he was working on Lin14 and on this lineage, on timing, and he saw they would talk to each other in the night and they would say, oh, the Lin4 sequence looks very similar to the Lin14 RNA in some part of the RNA. So they very quickly realized that Lin4 and Lin14 could bind to each other, and Lin4 controls the expression of Lin14, which is really incredibly elegant and carefully done genetics. I still can't get over it. It's so amazing. This morning I looked at those papers again and I was grinning like a kid. Okay, so they got the Nobel Prize last year. They also got the Breakthrough and Lasker Awards. But once it was discovered, for a while there was nothing happening. They just thought, oh, this is some worm-specific thing. But soon people began to discover these kinds of small genes everywhere. And they realized that this non-coding microRNA just normally controls and sculpts gene expression. If you have 100 transcripts, you can have a microRNA which comes in and blocks how it gets translated. Right? So the 100 transcripts, some of them are just silenced, some are taken out. So it can sculpt gene expression. And this turned out to be very, very, very important. This also has a lot of implications and applications because you can use it for diagnostics, like for cancer. If you have certain genes going up, going down, you can use this RNA-based technology to measure things, right? And you can take some of this pool, and if you can attach a probe to it, then you can sense what the levels of DNA or of the messenger RNA are. So this is again something done universally, controls a lot of gene expression in a lot of cases, including circadian rhythms, which is very important for us to be able to synchronize ourselves, and driven by primarily light in the environment. Okay, great. The fourth Nobel, making major contributions. And what were they studying? They were not looking for non-coding RNA. They were looking at some esoteric, how do cells, what time, timing of cell division, etc., etc. And they just came upon something which was very exciting, very new. Serendipity and curiosity careful experimentation, and persistence. I'll come to the final part of my talk, which has not gotten a Nobel Prize. But if you remember, Brenner was very interested in understanding the brain. And he thought the worms would make a nice, simple model. If you want to have a nice, simple model, you need a catalog, right? If you want to describe a plant, you will say it is this height, there are so many leaves, there are so many flowers, this shape, etc. So if you want to understand the nervous system, you catalog and describe the nervous system. And the way they did it, painfully, John White with Eileen Southgate and Nicole Thompson, who was a graduate student with Brenner and then stayed on after his PhD, developed computer programs in the 70s where none existed, and developed technology where none existed. And I'm almost done. Cut the entire nervous system, found the position of every single neuron and every single axonal processes. And that paper is called The Mind of the Worm. And it's a reference thing which people like us will go back and say, oh, what does this cell do? And I'll give you an example here. You say, oh, these are the synapses that it makes, and I'm not going to walk you through it. These are all the cells which make synapses with this. There are 2 synapses made with that, 5 synapses made with this. And this allows you to, for the very first time, get what is called the connectome. The fruit fly, this has only 302 neurons. The fruit fly has 100,000 neurons, and we just got the connectome this past year or 2. And we have done a 2-micron square piece of the human brain. This was done in the late 70s, published in 80s. We've had that information in the worms for a very long time. It is exactly that. It's the first wiring diagram of any nervous system. We know for all developmental stages now, we know for the male, we know for the hermaphrodites, we know how many synapses are made by each neuron and in which place. It's just an incredible wealth of information. Okay, and you always go back to it. Here, a movie that I made many years ago, where the neurons with which you can feel touch, okay, have been labeled. And these are places where neurons talk to other neurons, which you see shining brightly with a GFP over here, which is fused to a particular protein. So Marty Chalfie, who got the GFP Nobel Prize, was the person who worked out the first functional circuit when he was with Sidney Brenner. He said, how does the worm feel touch? The other way to ask is, how do you feel when someone comes and touches you on the shoulder? You know it, right? How do you feel that? And he figured that out by killing individual neurons and figuring out which of these 302 neurons was important for touch sensation in concert with the connectome. Finally, this is Corey Bugman, who not some while ago got the Breakthrough Prize, and she's got lots of other prizes, where she started when she was in Bob Horowitz's lab, looking at how do worms smell. She developed the circuit, identified the circuit for olfaction, identified that you can have a fixed circuit, but a lot of plasticity in the way you learn, where you sense, adapt, respond, and learn with your sense of smell. And she's done extremely elegant, seminal experiments to address these questions. So the question is, and I leave this answer to you, is C. elegans all it's made out to be? I think so. It's an extremely manipulable system, has a lot of tools going for it, and it has made major contributions to biology. I'm going to skip all of this, and I'll come to this last slide. Where does the success of C. elegans come from? I would argue that a charismatic leader like Sidney Brenner, with a tremendous sense of humor and certain amount of quirkiness, very bright, inspired many young people to adopt this model. And you saw this whole lineage, Martin Chalfie, Bob Horowitz, John Sulston, John White, all of them came and learned from him. A lot of people got their start, Victor Ambrose, Corey Bargman, Gary Ruvkin, in Bob Horowitz's lab as well. And a lot of work which was done was curiosity driven rather than merely hypothesis or results driven. So your eyes were always open for surprises. You did careful experimentation, you were persistent, and you figured out cool stuff. It was and still continues to be an intellectually engaged, open community where people share ideas, reagents, and just thoughts about everything without necessarily having ownership on it and say, we will not share this with you, which is still a fairly unique culture even within biology. And of course, what we did learn from the worm is that biology is universal. Many things are universal. So the findings that you have from using this organism is applicable widely. And finally, I'll end with another Brenner quote. And I think that's the true power of the worm. Need to acknowledge the great debt I owe to a large number of scientists who joined me in converting Caenorhabditis elegans from what was once regarded as a joke organism into a powerful experimental system it has become today. And I count myself very fortunate to be able to work on this organism as well as in the community that works with this organism. Thank you. I'm happy to take questions.
Sandhya, I'm going to provoke one question which you will not answer now. But since you said that it's curiosity driven and people keep their eyes open for surprises, after we come back from tea, maybe you can also share some of the surprises that you have had in your career with working with this tiny worm. But I think before we stop our live stream, we should look at if there are any interesting questions online. So, any interesting questions online?
So there's one question which says, what is Lin4 and Lin4?
Lin4 is so what is Lin4
and to know what is Lin4 and Lin14? So if found only, is it only found in elegant, whatever, or found in other species as well?
Lin4 is found everywhere. It's one of the first microRNA. Now microRNAs have names like MIR1, MIR147, MIR whatever, right? So that's a microRNA. Yes, Lin4 microRNA is found in lots of places, as is Lin14.
So Lin4 and
that's
Lin14,
just the worm.
Like Arnab or something.
Exactly. They're names given to mutants which had lineage defectives. That is, they had defects in either vulva formation and they also saw timing defects in cell division. So they call them lineage defects.
Okay. And then you may have answered these questions, but I guess it's probably a thing that fluorescent proteins are indeed important in biology, right?
Oh, super important. It just opened the door to doing a lot of cell biology and developmental biology particularly.
Okay. All right. I think those were the ones that were there over here. And with that, I think we should take a break. And online audience, if you still have questions, please remember that you can email them to us, outreach, outreach at tifr.res.in, and we will pass them on to the speaker. Now, audience here, don't go away. Don't worry, there's chai, and then we'll come back and there's more questions, etc. And with that, I think we can end the live stream and let's go outside, have a cup of chai and come back.
Okay.
Thank you so much,
Thank
Sandhya.
you. Thank you. And you guys can watch worms too when